Hello everyone and welcome to this week's lab where we will be looking at photosynthesis and different wavelengths of light. So this week you guys should have read and completed the lab background, or you should do that next. Then we're going to watch a lab simulation video and use the data provided to make some graphs. And we'll go into more detail about what those graphs are. And then you guys are going to complete your lab summary, upload it to Canvas, and then do a Flipgrid video. So to get started, we're going to go through, well, a lot of the stuff that will be more relevant to the in-person labs, but it's still good for you to know. But we'll be looking at why some plants grow better under certain lighting conditions than others. So light is going to be the big factor that we're looking at this week. Right. So in addition to the lighting conditions that we're going to be using, there are two major parts of this experiment that you need to be thinking about. The carbon dioxide production that you'll be measuring, and then the leaf pigments that are present in the plants that we're looking at. And the goal is really to describe how these pigments are linked to carbon dioxide production. So you'll see this in the, well, you may not see it in the lab video, but ultimately you're going to take a leaf. You need to make sure that they are about the right size. You want to be consistent across things so that you can actually compare them. Again, this is more procedural stuff. You want to obviously, like if you were in person, you would want to make sure that the leaf, the part that's actually absorbing the light, is facing upwards and able to do so. These are the lights that are used to collect the measurements that you'll be using. I guess don't worry about this. Again, more relevant for in-person. So this part is relevant. You're going to be using red, blue, and green lights for this experiment. And so when you think about that, rainbow gradient, you're looking at where these are falling on those areas. So the beginning, middle, and end of the visible spectrum of light. And then really important to this is the dark control where there's no light, so no photosynthesis is occurring, but the mitochondria in these plants are still active. And this might be a common misconception, but plants do have mitochondria. It's not just chloroplasts that they have. So they're still undergoing cellular respiration even when they're not photosynthesizing. So this will be what the data looks like, and you'll be getting the slope again. So similar to last week where you were looking at metabolic rates, you're looking at the rate of CO2 production. So not just the amount of CO2 present, but the amount that it's changing over time. And that'll be very relevant to your lab summary. So carbon dioxide sensors are what we're going to be measuring. If we could measure oxygen changes, that would be great, but oftentimes with these kinds of things and in papers that you may end up reading, you're going to just be looking at fluxes in carbon dioxide. But just because we're only looking at carbon dioxide doesn't mean we still can't make some inferences about what's happening in terms of plant growth. So there's a couple things to keep in mind that are going to really help you interpret these data. So photosynthesis uses carbon dioxide to make food for the plant, as you guys probably know. So it pulls in carbon dioxide out of the atmosphere. But if photosynthesis is not happening, then plants are going to be instead producing carbon dioxide. And this actually is happening all the time. Plants' mitochondria are always active, and they're always going to be producing carbon dioxide. But when photosynthesis is occurring, because they are both pulling in and producing carbon dioxide, the rate of carbon dioxide change in these chambers is going to change if they're photosynthesizing. So while in the dark we could have this trend line that's nice and a nice positive slope increase, when photosynthesis is occurring, because they're pulling in and putting out, that rate will probably slow down, or you would expect it to slow down or even become zero or negative. So really think about in terms of change in carbon dioxide, what's going on under these different light conditions that we're giving these plants. So let's think a little bit more about the photosynthesis part of this. We care about this because if plants are photosynthesizing more, that means they're making more food for themselves, which means they can grow more. So they're going to take in the light energy and they're going to store that energy as food, glucose, and then it also will produce oxygen, which is great for us. but let's look a little bit closer at that. So the color of light, it matters, right? And so a lot of times when we think of color, we're used to thinking of it in terms of paint, where you have these colors, you mix them together and they make, red and blue make purple, and we know that, right? But with light, it's a little bit different. You can see in this image that the plant is green, right? And so what that means is that that plant is reflecting green light and it's absorbing all of those other colors. If it was reflecting all those other colors, you would have a different color plant. But instead, because it's taking in part of the spectrum, what it's not taking in is what we see. So more absorption, those colors of light that we're not able to see, that's what the plant is using to make its food. And so more absorption of that light means more photosynthesis, and again, that means more plant growth. So the molecules that are actually doing the work in absorbing these wavelengths of light are the pigment molecules. And so there's a couple that we're looking at: chlorophyll A, chlorophyll B, and carotenoids. And so the plant that we're testing has a combination of all the three of these kinds of pigments. And so they're a little bit different in how they function. So in this one, we can see the carotenoids. They are, this graph along the x-axis is the color of light that it's absorbing, and the y-axis is how much it's absorbing. And so you can see that this one is absorbing a lot of the blue and a little bit of the purple light. And then chlorophyll A, it's absorbing a slightly shifted kind of light, more purple light, and it's also absorbing some red light. And then lastly, we have chlorophyll B, which you can see that dip in absorption in the carotenoids is right where chlorophyll B is absorbing a lot of that blue light, and it's absorbing a slightly shifted color of red, maybe a little into the orange. And so the color that's left, the color that's not being absorbed, is the color that's going to be reflected, and so that's the color that we see. And you can see that it's the green and yellow. So that's why we see this green color with these leaves. And this is kind of a more nuanced approach to looking at light. Again, like I said, most of us are used to thinking in terms of mixing. And so this software can use this concept of absorbance and reflectance, and it actually models what it would look like when you change the color of light. So here we have a yellow ball under white light, and if we shine a blue light on the yellow ball, what color would you think you would see? And so a lot of students are going to say, yellow and blue is green, so you should see a green ball. Or if it's a blue light, you should see a blue ball, or maybe the color isn't going to change. But instead, what we see when we actually change the color of the light is that we don't see the yellow ball anymore at all, because that yellow ball is absorbing every color except for yellow. Yellow is the one it's reflecting. And so if you're not shining any yellow light on it, instead you're just shining the blue wavelengths, that ball is going to absorb all of the light and it can't reflect anything, so it's going to appear black. So remember that the ball appears yellow because it's absorbing everything but yellow. And so if you're having problems with these concepts of absorbance and everything, you need to come talk to me, send me an email, and we can set up a time, or just stop by my office hours. So remember this extra part that we're asking you to include in your lab summary, this experimental design, and you have to describe your hypothesis, the two variables, or the independent variable and the dependent variable, and there may be more than one of each, and then your control group and your experimental groups. And so to go over hypotheses a bit more, your hypothesis in this week's lab should link light color, plant pigment, and photosynthetic rate. So not just the amount of carbon dioxide, because that'll be misleading, but the change in carbon dioxide. And so an example hypothesis is that plants photosynthesize faster or slower under blank light conditions because, and then your because should include something about pigments, because that's really the concepts that we want you guys to link. So based on your data, this would be part of your conclusions. Based on what you see and whether your hypothesis is confirmed or not, can you recommend a color of light that would be best for plant growth? And so again, please reach out to me. I've had some students come to office hours, and they've been more successful in their lab summaries. But I have also gotten a lot of comments about the lab summaries, and that you guys would like a little bit more clarity in what is being expected. And so we can do that. So here we have the rubric that is on Canvas. I pulled this right from Canvas. And so this is going to be how I go through your lab reports. And so I'm just going to go through the sections and explain what I'm looking for. So to start with, the quality of the summary. This one is very straightforward. Is it the right length, and can I read it easily? Is it grammatically correct? If it's around 300 words, and it's good sentences, then you're going to get 2 points for this one. Typically, students are losing points on this one because it's way too long. It should be about a page. Your figure can push it over 1 page, but roughly 1 page is what we're looking for. And so then we get to the introduction. And so there are 4 things that we're looking for here. You need to, and you can kind of think of the introduction as you're really the lab summary. You're starting very broad with your introduction, which is going to pull us into the experiment. You're going to explain the experiment, and then you're going to make it more broad again at the end with the conclusion. So for the introduction, we're looking for you to introduce the major topics. This doesn't need to be like reporting back the lab summary that you read. You don't need to give a ton of detail. You're not showing me that you understand photosynthesis. You're showing me that your experiment relates to photosynthesis and how. You need to explain why the work was done, like what are you hoping to get out of this experiment. You need to include your hypotheses, and your hypotheses need to be formatted correctly. And then you have to just you don't have to go into a ton of detail, just explain how the work was carried out. So those are the 4 things we're looking for, and then you can see you'll lose points if you don't include all 4. So then in the methods, if this were a full scientific paper, you should be able to reproduce exactly the experiment that you did just from reading the methods, but that's not quite what we're looking for here. Don't just copy and paste from the lab manual. You're going to use up a lot of your word count, and you're going to have less time to talk about other things that may be good. So you need to just explain the groups, so the experimental, the control groups that you used. Give a brief summary of the protocol. It doesn't need to be exhaustive, but you do need to go over the key points of how this lab was done, what type of data was collected, and then how that data was interpreted. In scientific methods, they'll explain the data they collected and the stats they used. But in this case, just explain how you're interpreting your data. So then for the results, here you need to, in text, so it's not good enough just to include a figure, you need to, in the text, summarize the trends in the data. So don't just explain your raw data or explain, or like give, an explanation of this group got this result and this result and this result. Like it needs to be the patterns that you're seeing, and they need to be correctly described in text. And in addition to this, you also need to have your figure. And so your figure, I should be able to interpret what kind of experiment was run just from your figure. So it has to be pretty clean. You have to have everything labeled, all of your axes labeled. I think students are having sometimes issues with what kind of figure you need to make. And this week we go into some detail about what figures you are supposed to be made in the lab summary, or not the lab summary, it's on Canvas. And so you really need to put some thought into this. Make sure that if it's a line graph, you have the equation posted. And make sure that the data makes sense to have a line graph. But again, I should be able to interpret this without reading anything else. It should just be a very clear figure. And so then we come to the conclusion. One of the main things I'm looking for is that you relate this back to your hypotheses. If you can go through and say what your hypotheses were, supported or rejected, like you're going to get points on this part. You need to tell me what it means, what your data means, and like relate it to a broader topic. So you can kind of think of this as relating back to your original questions and why we did this. And so conclusions can be a little tough. You have to really think about the experiment. Why would we have you run this? What biological concept are we exploring with this lab? And can you summarize briefly what your results can say about that biological concept? And so then you have your Flipgrid video. There's the 4 parts, like literally just check it off, write down what you need to do. And some folks I can see are reading off of like a card next to their screen, and that's fine. That's what we want. We want you to just be communicating to me how the lab went, what you got out of it, and stuff like that, just so that we can interact a little bit more. And so this rubric might change, and it might include the experimental design part in the future. You still need it even if it's not in this rubric. But just if you have any questions at any point in the future, don't just flounder on your own. Please talk to me. Please come to my office hours, send me emails, all of that kind of stuff. It's because we don't have the 3 hours in person. Your interactions with me are largely self-driven. You guys are going to have to come to me because I can't ask you questions in lab and interact with you that way. So I hope this helped clarify a bit what we're looking for in these lab summaries. And if not, then you know how to get in touch with me. And good luck this week.